Assalamu alaikum, dear student. Welcome all to online lecture class. I am Dr. Muhammad Munir Hussain, Assistant Professor, Department of Anatomy. Today, I want to discuss with you the basic concept of histology. At the initial point, we read the microscope. Thereafter, cell biology. So let us start. At first, you have to know the basic structure of microscope. That is, you see that in the above eyepiece, your eyes are placed in the eyepiece and see the slide. Thereafter, body tube. This one known as nose piece. Nose piece moving around, and nose piece fixed with objective. Objectives are four types here. Four magnifying power, four into magnifying power. Thereafter 10 magnifying power. Thereafter 40 and 100. That means when you place a slide, you can see by four times multiplying magnification by four objective. Thereafter 10 times, 40 times, and 100 times magnification possible by those objectives. So at first, slide placed and see in the four magnifying power, that you can visualize the whole area of the slide. And thereafter, you move the nose piece. You move the nose piece and fix the objective in 10 power. And if you want to need more magnifying slide, then you fixed it in 40. Now this is a stage. You placed a slide in this stage and fixed by two clip in each side. Thereafter, this is arm of the microscope. This is coarse adjustment screw. When you placed a slide, after moving the coarse adjustment screw above or below, you can fix the slide in approximate position by seeing the place your eyes in eyepiece. This is fine adjustment screw. When you place the slide in approximate position and focus the area of the slide, then you can use the fine adjustment screw for more clean cut view or image. In below the stage, here is a diaphragm to concentrate the light into the slide cover slip. And this is a point of middle round aperture or hole. From there, light can passes and concentrate through the cover slip area of the slide, tissue area of the slide. And this is light source. From here, the lights are passes upward. Here, switch present of the light. Here, this is base. So this is overall about the microscope, and you need to do practice in blank slide at first. Thereafter, use different histological slide and see under microscope. Now, cell biology. What is cell? Cell is the basic functional and structural unit, the smallest living part of the body. That means our structural and functional unit of the body is known as cell. Our body, actually all things are composed by cell. Two type of cell present: prokaryotic, eukaryotic. You already know difference between prokaryotic, eukaryotic. And prokaryotic present in case of bacteria and eukaryotic in animal cells. We discuss about the mainly eukaryotic cell or human cell, animal cell. The eukaryotic cell composed of two basic components, that is protoplasm. Protoplasm contains cytoplasm and more dense central nucleus. You see central nucleus, and Around the nucleus, different microorganisms present, and those organisms present in the cytoplasm, that is termed as protoplasm, and covered by a single membrane that is known as plasma membrane. Plasma membrane, that is the limiting membrane that envelops the cell, separating the cytoplasm from the surrounding structure. And its thickness is important. It's 7.5 to 10 nanometer. It's important for MCQ exam. Thickness of the cell membrane 7.5 to 10 nanometer under electron microscope. Structural component: protein 50%, that is peripheral protein that is situated in peripherally, and integral protein that penetrate the phospholipid bilayer. Here phospholipid bilayer showing, and it has two regions, that is hydrophilic and hydrophobic region. Hydrophilic means it has dissolved in water, but hydrophobic means it is phobic in water. Thereafter, few cholesterol, glycoprotein chain, glycosaccharide chain. A total image of fluid mosaic model. It's important for written exam. Drawing level fluid mosaic model. Phospholipid bilayer. Here a few protein channel present, that is integral protein penetrate the whole over the lipid bilayer, and peripheral protein situated surface of the lipid, and few chain present, that is carbohydrate chain, protein chain, glycoprotein, glycosaccharide chain. 1972, Singer and Nicholson proposed the well-established fluid mosaic model. So Singer and Nicholson 1972 discovered or well established this fluid mosaic model. Function: lipid bilayer lipid bilayer produce a physical barrier. Thereafter, selective permeability. Few particles can enter into the cell membrane and few particles cannot enter. Thereafter, electrochemical gradient and communication. Cytoplasm. Cytoplasm consists of many fluid components: cytosol, metabolic active structures, organelles, some protein component, cytoskeleton, and some deposit of carbohydrate, protein, fat called inclusion. So what are the cell organelles? Cell organelles are important for exam, and you have to describe it by two variety, that is membrane bound organelles and non-membranous organelles. So what are the membranous organelles? At first mitochondria, thereafter endoplasmic reticulum, thereafter Golgi apparatus, lysosome, peroxisome, and non-membranous organelles: ribosomes, centrosomes, microtubules, and microfilaments. So basic cell structure and ultrastructure of cell. It's important for written exam. At first you see the cell membrane, thereafter draw nucleus in the centrally, and attached or adherent to the nucleus rough endoplasmic reticulum. Why rough? It is because ribosome attached with it, and where ribosome is detached from the endoplasmic reticulum, it is known as smooth endoplasmic reticulum, and this is rough for ribosome attached with it. Thereafter, near the rough endoplasmic reticulum, Golgi apparatus present. Here, ribosome and rough endoplasmic reticulum or smooth endoplasmic reticulum synthesized nutrient like protein, lipid, and those substances are transported to the Golgi apparatus. Thereafter, Golgi apparatus can transmit through lysosome or vesicle, different vesicle, and those are transported through outside the cell body. And other structure you see mitochondria, centriole is a single, and when it is remain in right angle to centriole is known as centrosome. During cell division the centrioles are separated and goes to opposite pole in daughter cell. Here different tubular structure, microtubules, microfilaments that maintain the shape of the cells, and lysosome it has hydrolytic enzyme few, and it destroy the foreign particle. Now ribosome. Ribosome electron dense particle that is about 20 into 30 nanometer in size, composed of 4 segments of rRNA and about 80 different proteins. It has two subunits, a small and large, come together binding a mRNA strand, and by binding with mRNA strand, the ribosome or polyribosome can synthesize the protein. And you see the sequence here, a filament like structure that is mRNA, and here ribosomes, small subunit and large subunit are attached, and start when signal can come through this mRNA, the ribosome start to synthesize protein, and this protein when synthesized complete, it is separated from mRNA. Cartoon showing here the synthesizing protein by attached with mRNA. So I say that when multiple ribosome attached with endoplasmic reticulum, it is known as polyribosome or attached polyribosome, and when multiple ribosome remains free, it is known as free polyribosome. Now endoplasmic reticulum. Endoplasmic reticulum, I already told that two types: rough endoplasmic reticulum, smooth endoplasmic reticulum. Rough due to attachment of polyribosome, and endoplasmic reticulum remains adherent to the nucleus. Here, see it is adherent to the nuclear cell membrane or nucleus. Here, attached of polyribosome with endoplasmic reticulum and helps in protein synthesis, and that protein are transmitted through different site. Now, location of rough endoplasmic reticulum. You can see the different protein synthesizing cell present in our body, and uh, in the protein synthesizing cell area, the rough endoplasmic reticulum remains prominently, or huge amount of rough endoplasmic reticulum present there. Example is pancreatic acinar cell. Different pancreatic enzyme protein in like nature, and plasma cell also, and different other cell contain the rough endoplasmic reticulum. And smooth endoplasmic reticulum mainly present in the steroid secreting cell. We know that it synthesizes lipid, and steroid all are lipid in nature. So and adrenal cortex that produce different hormone, cortisol, aldosterone, sex hormone, all are lipid in nature or steroid in nature, present in the liver cells, because liver cell also synthesize different fat soluble vitamin that is also lipid. And the smooth endoplasmic reticulum abundant in lipid synthesizing area, that means liver and adrenal cortex, and it is present in the specialized form of skeletal muscle as sarcoplasmic reticulum. When we study about muscular system, we see that a special variety of endoplasmic reticulum termed as sarcoplasmic reticulum present there. Function in a single word: protein synthesis.